Hello everyone. So welcome to my live stream. I'm doing first time from my channel. So welcome guys. So let me see who are online. Oh, hi Hiten. Welcome. So I'm waiting for other people to join. Okay, so a lot of people are joining. Ishika, Raj. So good to see you. Yeah, it would be recorded. It would be recorded, but stay in live because you can interact with me, right? Yeah, hi Krishna, hi Pritam. So hi everybody. It would be recorded. It would be available again in the channel in a recorded format, but it would be better if you stay in live. One thing, guys, my voice is clear. Is it clear or is it finicky? Tell me, guys, my voice is clear or not? You're not hearing any echo or anything, right? So let me just cross check in my phone. Yeah, so it's okay, good enough. Okay, so we'll wait a little bit such that people can join. And good to see you guys. Many of you guys I know by your name. And so this is a continuous series. Okay, so this series would go on for whole August, and we'll pretty much cover the immunology concept, and immunology is my favorite, so I would really love to teach this thing, and we'll cover everything. We would leave no stones unturned. So every day we'll have these kind of sessions. We'll also have practice sessions together, so we'll have fun together. Okay guys? Okay, so guys, shall we start? So hi everyone, whoever is online. Yep. So shall I start? So Bhadra and Ishika, yeah, Gitanjali, so everyone is online, right? No, don't worry, don't worry. So don't have to worry. So we'll do it together. We'll watch this. So make sure you watch the stream till the end, because this class is very important. This is the foundation, because if we don't understand this roadmap, when we walk along this journey, we'll be lost. So today we have to be very patient, and we have to really listen to what I'm saying, and we have to think about it as well. And this would make the foundation of this whole understanding of cell, of the immune system, cells and organs. Okay, and I'll also discuss what type of questions can be asked, what are the concepts you need to learn, what are the thought process you need to bring. So these things are very important. Okay guys, so yep, shall we start? I think nobody would join anymore, so I'll start. Okay, okay, so what comes in your mind when we talk about the immune system? Say guys, say what comes in your mind when we talk about the immune system. I'll tell you what comes in my mind. So immune system, whenever we read immune system, we'll try to compare it with the defense system of our country, right? So see the similarities, because defense system has several divisions. Immune system also have several divisions, such as innate immune system, adaptive immune system. Now you can understand that defense system has like first line of defense, second line of defense. There are different lines. There are air troopers, there are navy, land troopers. All of these are there, right? So this is the similarities between the two. Not only that, there are many other similarities. For example, the navy commandos need to communicate with the air force, right? And the way they can do this is by walkie-talkies, radio communication, etc. Imagine the same scenario for immune system. There are tons of cells in the immune system, and these cells need to coordinate between each other, right? These are police, army, even sometimes NCC cadets. So anything that you can think about. Okay, so anything, and we'll try to compare it and play kind of Call of Duty game. So first thing we should ask that why. So the first thing that should come to our mind, that why there is a huge diversity of cells in the immune system, right? Any idea, guys? I mean, at least right now we can talk about 15 cells. So why there is a huge variety? Why one cell cannot do all the job? Because the more the number of cells, first of all, their development requires energy, their development requires tons of signaling. So each of these cell type need to commit to a lineage, right? So it's difficult. So hi Anup Kumar. No, so which integrated PhD or PhD? If you are saying about TIFR, those are several days, okay? Different days. For PhD, there is a different interview, integrated PhD is a different interview, okay? So, I mean, at least there are so many different cell types. So already Bhadra has answered that the reason for so many cell type in the immune system, because the threats are diverse, right? So the counter mechanism should be diverse as well. Agreed, guys? But more the cells, more is the hassle, right? Because you need to build up a communication between all of these cell types. For example, let's say there is an attack, right? And now if you want to counter the attack, the army rangers need to really make a plan that, okay, so we'll go first, then you guys come when we signal, or if we fail, then you guys go. So these kind of first line of defense, second line of defense, backup, these kind of things has to be laid in the immune system as well, right? Otherwise there would be miscommunication, there would be a burst of response, and there would be chronic inflammation, right? Sometimes this happens in our body. Some pathogens really confuse the immune system, and all of these immune system, majority of these immune system kind of act in a non-specific way, right? So here comes the second part. So hi Neha, welcome to this live stream. Neha, hi Soumya as well. So there comes the next point, necessity of so many cells. So immune system need to be specific, right? Agreed, guys? Needs to be specific, right? Because without specificity, there could be risk of self-destruction mode or something like that, right? Yep. What do you think the third most important aspect about the immune system? I mean, okay, we can say that they need to communicate with each other, right? That's important. They need to communicate with each other. And the way they communicate is cytokines. Right? So cytokines are the way of communication between them. Do you guys agree that immune system need to be temporally coordinated? So the players of immune system have to be temporarily coordinated. Right, such that they have an idea that who is going first, who is fighting and everything. So this point is very important, that cytokine is the way by the immune system is communicating with each other, right? Now cytokine is a very big term, it's an umbrella term, right? Now let us try to understand the immune system, how is it developing, where do you find the immune system, etc, 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 right? So let's talk about that. So here you can see an individual, nice looking individual, handsome one. So let's try to talk about his immune organs. Okay, first of all, the most important component is the big bones here, right? Let's say the humerus. Then all the big bones have bone marrow, right? So these are the immune organs that we are pointing out, right? We'll talk about bone marrow later on, but femur, humerus, all the big bones have these kind of quite a lot of bone marrow, and bone marrow turns out to be very important for immune system. Imagine how scientists really figured out that bone marrow plays this role for immune system. Any idea how scientists can get a clue that bone marrow is the key for the birth of the immune system? If you take out bone marrow, there could be consequences. So how does scientists get to know about that? Any idea how they can possibly do it? No, B cells, let's say we are at a time when scientists don't even know about B cells. They saw that few type of pathogen body can encounter, body have some defense response against these external threats, but they don't know that how body can do that. And later on, eventually they figure out that bone marrow has a potential role and it is the birthplace for the immune system. Okay, so Saurav Sharma said that they extracted it. Okay, that's good, but think about it. When we are talking about human, extracting the bones is difficult, right? Like exciting the marrow is difficult, right? Yeah. So, yep. Think about the clinical setup, right? Think about the clinical setup. So if you have a clinical situation where due to some infection the bone marrow is gone, or due to some irradiation bone marrow is gone, or due to some cancer bone marrow is not functional, then what happens? That individual would be severely immunocompromised, right guys? So from those point of view, people had an idea, or especially doctors had an idea about the immune system's origin. And later on there is a big story how the immune system was discovered, but I'll tell you in some other day. Let's just focus on to our understanding. So apart from these bone marrow, just above the heart there is something known as thymus. Okay, now okay, so let us draw the thymus in a better way. Okay, so let us designate these things. For example, bone marrow, they are the birthplace. Okay, okay, thymus is the training school. Thymus is the training school for T cell. Okay, great. Now let's talk about some other ones. For example, look at these ones, the things that look like vein, they are actually lymph nodes. Okay, so these are lymph nodes. So if you think about lymph nodes, what are they? Okay, so you can think lymph node as army base camps. Okay, because lymph node is a region when exact, or office or anything like that. So you get trained in some other place, in school or college, something like that. So it's like a training place for the T cell, and then you go to your workplace. So the armies would be recruited into the bases where they would encounter the pathogen maybe. Now, so this is very important, right? Lymph node. Then comes spleen. So here is spleen. So spleen is actually another army base camp, but a big army base camp. You can think about the headquarters of all of these army base camp in our body. Now spleen is pretty big and has quite a lot of immune cells. We'll talk about all of these organs individually. Okay, we're back again, no problem. Okay guys, so let's talk about the development part, right? Please stay with me and we'll talk about the development part. So this is very important. The most important part, we are not there yet, but we are still doing a little bit of understanding of this part, okay? So you guys are here? All of you guys are here? Do some gestures guys such that I know that you guys are here. Okay, look at it. Now inside the bone marrow, which is the primary lymphoid organ, primary lymphoid organ means it's the birthplace. Primary lymphoid organs means it's the birthplace of the immune systems. So both T cell, B cell, or let's say, let us put it in this way. Both the adaptive or the innate immune components, both are present or both are born in this bone marrow, right? So if you take a cross section of the bone, what you are going to see? You are going to see few things, few addresses, few colonies, let us say, right? Yeah, I'll share the whiteboard file later on. So there are few addresses. Something is called endosteal niche and something is known as the vascular niche. So the endosteal niche is actually residence of the hematopoietic pluripotent stem cell, or mainly progenitors, right? Now vascular niche is actually an address for the cells of immune system which are differentiated. So this is very important, right? Because see how nicely this place or this region is segregated. You have a place for the immature cells or the cells which have renewal properties, and you have a place for all the differentiated cells which are going to be matured. They come near to the blood vessels because they need to move around the body, right? And many of these blood cells are actually taking part in this immune response, so they need to be coming close to the blood vessels, right guys? Agreed? Now, so this is important to know the endosteal and the vascular niche, right? Now let's move on to really the bone marrow to see what type of cells are there and how they are produced, okay, and how they are born. So let us take a big picture of the brain here, sorry, big picture of the bone here, not brain. Somehow I'm too much fond of brain and saying brain all the time. Yeah, so here is our bone, right? So inside the bone there is stem cells, and I hope you guys know what are stem cells, right? So they are hematopoietic pluripotent stem cells. Otherwise they are also known as HSCs, or hematopoietic pluripotent stem cells. So I hope you guys understand what does pluripotent means. Now pluripotency is defined here to say that these stem cells can give rise to all blood cell types, all of the blood cell types, okay? So from the hematopoietic pluripotent stem cell you would have progenitors which are a bit more differentiated than these hematopoietic pluripotent stem cell, okay? So these progenitors are broadly divided into two categories, okay? And this is very important, and there are two different lineages here. They are bifurcated in two different lineages. So one would be lymphoid progenitor. Then the second would be the myeloid progenitor. So directly this is very important from any exam point of view, because they ask about like, okay, these cells, they are derived from which type of progenitors or which type of lineages. This is a very common question. We'll talk about it tomorrow. But look at it. In the myeloid progenitor, I mean, if I say broadly, I'm very broadly speaking, but myeloid progenitor, this gives rise to components of innate immune system. Okay, whereas lymphoid progenitor gives rise to components of adaptive immune system. And you would be saying, what is adaptive, what is innate? Anybody can you tell me? Do you guys have a very good idea about what is innate immune system and what is adaptive immune system? Any idea on that? Okay, so we'll talk about innate and adaptive system as we go along, but let me tell you in brief that innate immune system means they are already, this kind of immunity is inborn, like it is already there, right? But this adaptive immune system, you need to gain this immunity, and it is only gained when you have sort of like exposure to the pathogen. So I'll tell you this is very similar to have a smartphone. You have some default apps like Google Chrome or Google browser, these things, and you have some apps that need to be installed, right? So you can imagine it in this way. Now there are many debates about how they work, how they can possibly work and all, right? We'll talk about it as we go along in this course, okay? So stay tuned with me, it would be really interesting for you guys, okay? So let us try to understand. So what happens after this? So lymphoid progenitor, myeloid progenitor. So from myeloid progenitor, many other cell types would be generated. For example, blood cells. For example, blood cells means RBC and several WBCs, and also platelets would be generated from the myeloid progenitors. Okay, but let us not talk about WBCs or platelets today, because we are more focused towards the components of the immune system. So we'll see what others are produced. For example, they will produce monocytes. They will produce monocyte progenitors actually. They would produce basophil progenitors. They would also produce neutrophil progenitors. That means they are not mature yet. They are kind of lineage committed. I mean, they have to become these, but they are kind of immature, right? So, neutrophil progenitor. Other than that, there would be eosinophil as well, right? So all of these things are actually in a progenitor format, right? I'm not drawing all the blood cells or anything. What would happen, guys? All of these would be secreted. All of these would be secreted into the bloodstream. Okay, so let us take some questions. So innate is default. Yes, that's true. Adaptive is evolved, right? Yeah, that's true. That's true. Okay, how NK cells are produced? We are coming to that, okay? So we are coming to that, don't worry about that. Okay. So, exactly, so that's a good question. But let us deal with these things. Apart from that, there is another cell type which is mast cells, okay? So mast cells are also a product of these lymphoid progenitors. We'll talk about mast cell later in context of allergy. So we'll talk about all of the function of these cells, but let us look at the lymphoid progenitor lineage, okay? So from the lymphoid progenitor, the major 2 cell types or 3 cell types that would be produced is these ones. So first of all, there would be T lymphocyte, T cells. This is like T cell progenitor, I would say. T cell progenitor or precursor, anything. There would be also B cell. So it's B cell progenitor or highly immature B cell. So I'll say it's B cell precursor, okay? And then also another cell type which can be produced from here is basically the NK cell. So right now, if you see many cells, so how do you differentiate between all these cell types? What are the ways by which you can really differentiate between all these cell types? Any idea, guys? What experiment would you do to differentiate between all these cell types? One possible way is to do FACS sorting, right? Because FACS sorting can really sort these cells. We know all of these cells are not similar in size, neither they are similar in granularity. Some cells are more granular, such as the granulocytes. Some cells are less granular, which are agranulocytes. And these cells have different kind of surface markers. So first of all, morphology, and second, the surface marker, right? So question was how immune cells could be sorted. So quick answer is FACS, right? Flow cytometry based method. Separate them based on 1, morphology, 2 is basically their surface expression, surface marker. So people often ask me that there are so many surface marker, how do you know what to do and all of these things? You really don't need to know. There is actually a catalog. So whenever you see some cells with some kind of marker having CD, XYZ, you just go to the catalog and look at that catalog that this thing matches with what, right? So that is how it is done. Not, you don't need to remember, but what you need to remember is the overall framework, that how things are happening, okay? So this B cell precursor would be secreted, T cell precursor would be secreted, NK cell would be also secreted, okay? So this NK cell has a unique signature, okay? They have signatures. This is called NK 1.1. This is a unique surface marker which is exclusive to NK cell, not present in any other cell types. Actually yes, but let us just not go to very complication right now. So NK cells have NK 1.1. This is their molecular signature. One way to identify is to identify your signature. So this is how you can be identified. Just like you, cells can be also identified with their molecular signatures. Now, so NK cells are there. Okay. Now guys, you have to understand the B cell precursor are in the blood, T cell precursor in the blood. Also these monocytes, basophils, neutrophils and eosinophil progenitor, all of them actually ultimately secretes, I mean all of them, they move towards the vascular niche. Remember there was there is a endosteal niche and there was a vascular niche. So endosteal niche was closer to the bone, right? So you can imagine this is kind of a niche, a broad line that is dividing. This is like a niche. It's a very awful way of saying it, but yes. So right now these monocytes would be eventually secreted in the bloodstream, right? But eventually monocytes, when they are in a circulatory phase, they are monocyte, but they can move and infiltrate the tissue space, right? Now in the tissue space, monocytes are no more monocytes. They get differentiated, they change their morphology, okay? Monocytes change their morphology and they get differentiated. Differentiated means you can imagine it as a maturation process and they become now macrophages, shown as Okay, so monocytes are actually precursors for macrophages. So monocytes in circulation and sort of like macrophages in tissues. Okay, so the lineage is same, the birthplace is same. For example, the monocyte is like your teenage, and right now these macrophages are right now your 20s. So you can imagine it's the same person, same lineage, but now their morphology, their characteristics are altered. For example, you would have more rough voice, more aggression, these kind of thing right now, rather than when you are a child, right? Lymphoid progenitors can also be macrophages. No, that's not possible. Like lymphoid progenitors does not give. Macrophage is coming from the myeloid lineage, but I'll tell you something which is very interesting about both of them, and which you need to know is something like this. So from both lymphoid and myeloid lineage, there is one interesting type of cell. Okay, this cell is known as dendritic cell. So there is a huge confusion in the community that dendritic cell becomes to which kind of lineage, lymphoid lineage or let's say in the myeloid lineage, and they have kind of signatures for both. So lymphoid lineages and myeloid lineages, right? Yeah, so these are the dendritic cells, let us call them DCs. So they can be derived from both. Okay, so what Soumya is asking, that what are the, so I have already said that there are specific surface markers. These surface markers could be signaling receptors, these could be adhesion molecules, even sometimes these could be glycoproteins. Okay, so as we move along this course, we will go to this nitty-gritty details. Okay, so guys, then what happens? Then your basophil, which was in a immature state, that also gets secreted. Neutrophil, which was very immature, right now gets matured and gets secreted into the bloodstream, right? And these would be circulating all around the blood. These are circulating. Okay, dendritic cell, NK cells. Dendritic cells, right? So let me tell you two things, that these dendritic cell and the macrophages, they are very important component of innate immune system. Okay, so these dendritic cell and macrophages, both DC and macrophages, are important components of innate immune system. Okay, so they are very important components for innate immune system. Now both DC, both the dendritic cell and macrophage has pattern recognition receptors. Okay, so I'll tell you what pattern recognition receptors are. Do you guys know what is pattern recognition receptors? So they both have pattern recognition receptors, right? Such as toll-like receptors, right guys? So they have toll-like receptors. Yeah, mast cells are also circulatory, yes. So they are in a precursor format, but when they leave the circulation, they get differentiated and they work what they want to work on. They are mostly involved in allergic response or hypersensitive response, and they lead to vasodilation and many other aspects. But we'll talk about mast cells later on, especially when we talk about the type 1 hypersensitivity. They both have toll-like receptors. Then both of them present antigens on class 2 MHC molecules. Okay, so about all of these things we'll talk when we talk more about the dendritic cells and macrophages. These lecture series is in a way designed that each day we kind of link what we have learned in the previous day. So that is how a sound concept can be generated for you guys. Okay. Yep. So first of all, what are the function of all of these things? And what I have not written right now, what we should have written, that all of these cell types, both dendritic cells and macrophages, they secrete quite a lot of cytokines. Okay. So okay, since we have talked about it, let's talk about the dendritic cells and the macrophages. Okay, in a bit more details. So let us draw the DC. So do you know why these cells were known as dendritic cells? Because neurons were well described by Santiago Ramon y Cajal, and people saw these some cell types which are present in the immune system. They have these nice dendrite like projections, making them look like a neuronal projection, and that is how the name came, the dendritic cells. They have these processes which are dendritic cells. Now what are these processes and why they are important? So looking at these processes, one can speculate that these cells have phagocytotic role, and these processes are dynamic. That means they grows and shrinks, they try to engulf pathogen, they actually can sample for pathogen with these long processes. So that is how these processes are very important, and the nomenclature is kind of appropriate, right? Everybody agree to that? So let us try to understand what are the most important points about the macrophages and the dendritic cell. So let us draw a macrophage as well with the dendritic cell and try to draw the similarities and the dissimilarities. Okay, so this is a macrophage. Say macrophage, and I'm going to say dendritic cell. The similarities between both of them, obviously you can see their morphologies are different, right? Differ in morphology. Okay, but so we'll talk about the similarities and dissimilarities. Okay, any similarities that you guys can think of? Both of them are APCs. Both can recognize pathogens. Via these pattern recognition receptors, right? Now both of them present their antigens that they have detected by class 2 MHC molecules. So very good point. So I forgot that, Navani, thanks for Hiten, actually not Navani, but thanks for telling me. So what they do, they are phagocytotic. So both of them takes part in guys what? In cytokine production. Both of them secretes cytokines. These cytokines are inflammatory in nature, many of the cases, but there are many other cytokines that are important. Remember, we talked about cytokines and we told that these cytokines are actually the communication signal. So let's come to the differences. They differ in morphology, they differ in half-life, half-life means how much they are staying, like their longevity. So dendritic cell turns out to have more lifespan than the macrophages. The reason is slightly unknown, but motility, so they differ in motility. Dendritic cells are more motile. Motile means they can move, okay? Whereas macrophages, they could be resident macrophages or they could be also mobile but less than dendritic cells. Dendritic cells are highly mobile. Other than that, most of these properties are pretty much similar. Both of them have class 2 MHC molecules. So both of them are not only antigen presenters, but they are actually professional antigen presenters, okay? So any cell type that presents microbial or any kind of pathogenic peptides on class 2 MHC, those are actually professional antigen presenter. But apart from that, this is a far-fetched thing. The first thing that they need to do is to recognize the pathogen. If they can't recognize the pathogen first, then how they would start and displaying the peptide. So let's say we have a bacteria here, okay? So these bacteria have flagella, they have cilia, right? So some components of the cilia or flagella can be actually detected by specific TLRs. These are known as toll-like receptors, which are one kind of pattern recognition receptor. We don't need to know the exact details today because tomorrow we are going to deal about that, okay? So TLRs recognize, okay? So basically PRRs helps in recognition, right? So they kind of help in recognition, PRRs. Once they can recognize, they would activate the macrophage. Now macrophage would, after getting activated, macrophage or dendritic cell, right? After getting activated, both DC and macrophage can secrete cytokines, right? This is very important because imagine a situation. They know that they have phagocytotic power, that they are not very specific and they don't have that much of killing power. So what they are actually like a police force. So who can engage with the enemies but also have a fear inside that. So what they need to do, they need to call for backup. And these cytokines that they are secreting are actually saying the immune system, other components of the immune system, that something wrong had happened, please give us backup, we need more backup. Not only other components of the immune system, but same kind of cells, let's say macrophages or dendritic cell, they call them and they look for backup, okay, such that they can survive and they can kill the pathogen, right? So this is very important. Not only they are a mode of communication, but they are also a mode of survival in this case. We'll talk about it more later, okay? So right now we have looked at dendritic cell macrophages, which are very important, okay, innate immune system components, cell types. Now let's see what we are going. Now we go to the next phase of our understanding. So we zoom in here again. So where are we? We are right now in the blood circulation. So in the blood circulation, we are going to now follow the T cells, okay? Now we are going to follow the T cells. The T cells, they are now in the blood circulation. Imagine this blood circulation is continuous, though it shows it's not continuous. But these T cells eventually goes to where? They goes to thymus, okay? So I already told you that thymus is the training school, right? So let's see what type of training they get in the thymus. So in order to do that, we'll take a cross section of the thymus and try to look at it, okay? So this is the cross section, and let us look at a bigger picture, a bigger view of it. We are zooming into a tiny portion in this cross section view, which is squared, right? So this is basically thymus. Where thymus is located? Just above your heart. Now guys, this thymus gets degenerated in older individuals, and that leads to problem, that leads to weakening of the immune system. Okay guys, so that leads to weakening of the immune system. Now tell me guys, do you know the story that how people figure out that thymus was very important for adaptive immune system? Any idea guys, any idea? There was a funny experiment, and with a very funny name. These cells are known as, okay, these are training camps. So they produce T cells, both T helper cells and cytotoxic T cells. But this idea came from a mouse, a transgenic mouse line, which don't have the thymus. Due to some reason, the thymus is ablated, like the thymus is not there. And those mice don't have any fur. That's a secondary consequence, but these mice are very much, what should I say, susceptible to kind of pathogenic attack. In simple words, their immune system is kind of non-functional. So they have to be grown in a very sterile situation, otherwise they would be totally dying. So then scientists think, okay, thymus has something to do, because if you remove the thymus, immunity is compromised, right? So let us talk about what happens inside the thymus. So already bioaddiction by Noman. So I hope your name is Noman. So Noman is saying there is negative selection, positive selection, many mouthful of things right now. But let us not, but we'll go when we talk about the T cells in details. So right now we'll try to think what happens to these T cells and how does they come from. So they were moving into the bloodstream, and there is something in the thymus which is known as high endothelial venule, okay, HEV. So these T cell precursor, or these immature T cells, okay, so these are immature T cells, right? So they sort of come in with the help of high endothelial venule. Now from there, where do they go? So they kind of migrate towards the peripheral region of these thymus. So you are looking at the cross section view. I hope you can relate which view you are looking at. So this is the internal side where I am pointing right now, and this is the external side. So this is the external side, okay. Right, and this is the internal side. So now guys, these structures are actually divided into specific regions, okay. So I'll tell you what. So first, they are subdivided into several regions. So first region is known as, okay, I think it's back again. Sometimes there is a problem in the stream. Okay guys, now look at it. So in this external most region, they move, and they not only move there, but they also divide there rapidly. They increase in their number, okay, in the subcapsular region. And these are, oh, what is HEV? HEV stands for high endothelial venule. So this is actually just a bloodstream, I mean a pipeline through which the immature T cells come in, simple as that. Now these are known as DN thymocytes or DN T cells. So these T cells have their T cell receptor, but they don't have, guys, am I audible? My stream is doing a little bit problem, but I think it's okay. So these DN T cells, they are CD4 negative, CD8 negative. So CD8 and CD4 are actually signatures of different type of T cells. But apart from that, there are important components sitting there, such as the thymic epithelial cells or TEC, okay. They are thymic epithelial cells. So there are many of them, okay. Now these thymic epithelial cells, what are they? They are teachers, okay. So eventually when these double negative T cells interact with these thymic epithelial cells, they become double positive, okay. Means now they are both positive, CD4 positive, CD8 positive. Then these DNT cells goes and further interact with other thymocytes present in the corticomedullary junctions, okay. So look at these, there are other ones, okay. Now if it recognize MHC one bind peptide, then what would happen? It would become T cytotoxic cells, which have the basic thing, T cell receptor, but CD8 as a co-receptor. That is one way. The second way is if they interact with class 2 MHC, okay. So if they interact with class 2 MHC, which I am determining here with this blue depiction, now they would become a T helper cells, okay. Both of them have the TCR in common, but now this is CD4 positive but CD8 negative, okay. So they have to take some choice. So already you know, so look at it. So imagine it to be a school, right? So these are like maybe the primary school, so they read all the subjects. Now as they go along, they are getting bit specialized. They are interacting with the teachers, so obviously they have one kind of motivation, motivation, either choose science or choose the art stream, right? If you choose science stream, you have to stick with science stream, you cannot fall back to arts. If you have to choose the art stream, you cannot fall back and go to science stream. So this is how their streams are specified. Either they become CD4 T cells or they become CD8 positive cytotoxic T cells, helper T cells or killer T cells. And this whole thing is directed by whom? By their teachers. The way they interact with these cells lead their differentiation into different populations, right? Either cytotoxic T cells or the helper T cells. So this is the overall summary that is happening in the thymus. Now let's say they become the T helper cell and the T cytotoxic cell. Next is what? Next is what? So right now these two guys have a degree, they are certified. These are certified by their thymic teachers, right? That means they have a degree right now, but they have not really gone to office. That means they know how to kill. They are army rangers, so they know how to use the gun or how to do it, how to press the trigger, how to use a grenade, everything. But guys, what they really don't know is how does an enemy look like, because they have not encountered the enemy yet. So they have done mocks, but not the enemy yet, right? So still both of them are now termed as, naive T cells, naive helper T cells. Either they are naive helper T cells or they are naive cytotoxic T cells. So everybody understood this part, that either they are killers or they are helpers. Both of them are very important. In army, only the front rangers are not only the important component. In army, the people who are doing the intel work or the intellectual work or sitting behind the machine, giving their intels or controlling the drones, they are also very important, right? So both the components are very important, but still they are in a naive state. They are not mature. Then how they would mature? So I hope you guys are not feeling it boring, right? Do you guys feel it boring? Then I would discontinue or I would continue the map. A little bit more, let's say 15 minutes more. It depends upon you guys. If you're finding it interesting, then I'll continue. Say yes if you're finding it interesting. The naive T cells need to be deployed into the army base camp. That is, in our case, it is lymph node slash spleen. We cannot ignore the spleen, right? Very nice question. So we'll talk about it. So your question is like, is there any bias for these teachers? Like, is there the students which are taking science stream or art stream, they're same, or some factor is guiding this? So I'll hold your breath, and also people think about it, whether there are similar numbers or there are dissimilar numbers. Is CD4 cells more important than the CD8 T cells, or both of them have very important importance in terms of immunity? So both of these factors would determine that they are numbers, right? I'll give this answer in next to next day. Okay, great. So guys, we are continuing. I'm thinking of continuing. I hope you guys are okay to continue. Okay guys, so now we had what? So after these discussion with you guys, what we know that right now in our hand we have T cells, okay, yeah. So we have T cytotoxic and T helper cell, but both of them are naive, inexperienced. Let me put it in this way, inexperienced. So both of them would now move to the circulation, and they would move along the circulation, they would circulate, and eventually they would reach an important place, that is nothing but the lymph node. So let us zoom into the lymph node to understand the situation. So lymph node, you can see here there are specific blood supplies to the lymph node. The artery takes the blood, and this should be opposite, but this is somehow not opposite. Yeah, arteries and veins. So you can see the cells either come through these blood supply, they comes in, they can go out with these lymph connectors, right? Now this is like an army base camp, right? This is the lymph node is equivalent to an army base camp, right guys? Everybody understood this part? So if this is an army base camp, there should be specific areas where you would find the T cells, where you find the B cells, right? So basically you have barracks inside, so you have specific regions. So this region is known as follicle, B cell area, okay, and this region is known as paracortex, that is T cell area. So you can understand both of them are there inside the lymph node, but there are distinct regions where T cells and B cells reside. For example, these navies would prefer to stay with the navies and they would sort of stay in a different barrack than these land troopers. They would also stay in a different barrack. And what would happen, guys? They would sort of interact. They can interact and they can communicate and activate each other. They would say, okay, now you should go to the war, and this is the way they can plan together. Okay, guys? So this is happening with cytokine, and which we would learn in T cell and B cell activation subsequently, not today itself. Okay. So now, guys, let us move back to a big picture of these. A big picture of these lymph node. Okay. Yeah. So let's talk about the big picture of the lymph node. Okay. So right now we have already looked at there were dendritic cells, right? Remember, guys, there were dendritic cells. Dendritic cells were there near your body surfaces, near your first line of defense. So they are just like a patrolling police officer near the border. Okay. So these dendritic cells. Now, whenever dendritic cells find something wrong and they would present it on class 2 MHC molecules, what they would do, guys? Dendritic cell would move towards this army base camp as if there is some kind of attack happened. They cannot control the situation entirely, so they are going to the army base camp for seeking help. Now, obviously, in our army base camp, we know that there are specific barracks for T cell and B cell, right? So in this barrack, there are B cells, right? So here in this barrack, what would you find, guys? You would find B cells, naive ones. Right? Naive ones. So B cells come to these barracks from bone marrow. So they don't have a training school separately. They train in the bone marrow itself. We'll talk about B cell development later on. But T cells come from these thymus and they move to the lymph node. So T cells, especially the T helper cells, would be found here, right? In the paracortex region, and they have CD4. So now what happens, guys? This dendritic cell comes here, and we are not going to talk about the specific interaction today because this is just an overview, not too much details. So these dendritic cell would sort of interact with the T helper cell, right? This interaction between T helper cell and dendritic cell, they would make the T helper cell active. Okay. Now, once the T helper cell is activated, it can now influence the B cell, right? So there is like a chain reaction. When the B cell is activated, B cell is no more like a B cell. So B cell change its shape and size. So B cell now becomes antibody secreting plasma cells, so which are secretory factory for antibodies. Okay, so they basically secrete antibodies into the circulation, and this is component of humoral immunity. You might have heard this term, humoral immune response and the cell mediated immune response. And we always say humoral immune response, that means it's antibody mediated, and it's a cell mediated response, then it would be cell mediated like T cells. So we'll talk about the bits and pieces of humoral and cell mediated immune response, but later on, not right now. Okay, guys? So everybody understand what happened in the lymph node? So everybody understand that what happens? So we have not talked about the B cell to plasma cell. So from B cell to plasma cell, there are several processes. Okay, there are several processes. So these B cells, they have these receptors. But imagine these pathogens are changing every day. So these B cells also need to make very high affinity receptors, high affinity antibodies against the pathogen, and the way it is achieved is by Okay, this is by somatic hypermutation. Okay, somatic hypermutation. So what is somatic hypermutation? We'll talk in a different class. We have a class for that. But anyway, so the cells which have very high affinity, okay, they would be accepted, otherwise the other cells would be rejected. So the cells that would be accepted goes to next round. They matures in terms of affinity, okay. And finally, you know that these B cells have membrane bound IgM and membrane bound IgD. These are isotypes. What are isotypes? We'll talk about it. Now what happens, guys? There would be class switching. Class switching. And after that, it can become a plasma cell which is secreting different type of antibodies, okay. So this journey from a naive B cell to a differentiated plasma cell is quite long. Several aspects have to be taken care of during this journey, okay guys. So this is very important. I hope everybody can understand that, right guys? Can you guys relate? Okay, so we looked at what happens in the lymph node as well. So now guys, what do? Now we move to other tissues such as spleen. So let us try to understand what is the importance of spleen. So where spleen is situated? Just behind the stomach, okay. So spleen is the biggest secondary lymphoid organ. So what are secondary lymphoid organs? Where immune response starts, and the primary organ is where the immune cells are born, okay. So here the immune response starts, so the cells starts fighting against the pathogens, so the cells get activated. Now there are many different, so this is a cross section of the spleen. So you have splenic artery, you have, yep, this is Yudhbhumi, great, and there would be a vein, splenic artery, splenic vein. I'm not naming them, but what is very important, these artery associated areas are known as red pulp, so that are rich in macrophages. And so spleen has two broad part, okay. One is, let me write it in red, so red pulp, which has blood cells, I mean RBCs generally, and then there would be macrophages. Whereas there is another thing which is known as white pulp. So white pulp has two regions, okay. Peri-arteriolar sheath region, pulse, okay, and there is another region which is follicles, okay. So this peri-arteriolar sheath region are region for T cells, whereas follicles are for B cells. So again you see in this big army base camp as well, okay, in this big army base camp you also have differentiation in terms of their residence. They have different barracks. They have the B cell barrack and T cell barrack. Now let me answer the question, is memory cell derived from B cell? So the memory cells can be derived from B cells and T cells both, not only B cells. So there are memory B cells, there are memory T cells. I have a dedicated class coming for that, so we'll talk about it later on. But right now, today it's the introductory class of this course, so we are talking about the basic stuff. So now, after learning all of this, if I ask you what is the biggest importance of spleen, do you know what is the biggest importance of spleen? So spleen is very important. Spleen is very important because it traps the blood-borne antigens, let's say derived from bacteria. I mean, mostly these antigens are actually bacterial exotoxins, such as Pseudomonas aeruginosa, or let's say Staphylococcus pneumoniae. These things are there, right? So these specific bacteria derived antigens can be found to be in the blood, and they sort of get detected in the spleen. Okay, so that is why spleen is very important, because lymph node cannot trap blood-borne antigen. That makes spleen so special. But if you do a splenectomy or remove the spleen, it really does not matter that much. But only problem is it makes you more susceptible towards bacterial infection rather than other type of infection. Right, guys? You understand? Now, apart from all of these tissues, there are many tissues such as gut associated mucosal tissues, or GALT, or let's say mucus associated tissues known as MALT. So these are kind of what should I say, these are temporary sites and not exactly army bases, but they are kind of like a checkpoints. Okay, they also help in the immunity because they also have T cells and B cells, limited number, not as many as the lymph node or let's say the spleen, right? So they are only for temporary aspects. Okay, so I didn't understand, Saloni, your question. So when you remove the spleen, you are more susceptible towards bacterial infections, because bacterial infections are exotoxin types and they are mostly actually soluble in blood. They're not drained in lymph, whereas the tissue ones which are present in the tissues, they can be drained via lymph, and dendritic cell can be taking them away, right? So these are the very basic overview. Now as we move along, we would see T cells have so many varieties. So whenever we talk about these, okay, so we say common lymphoid progenitors, CLPs, okay, common lymphoid progenitors. So these common lymphoid progenitors can give rise to T cell and B cell as well, right? They can give rise to T cells, but they can also give rise to B cells. Now question is, why does a immature T cell suddenly don't decide to be a B cell? Any idea, guys? Why don't suddenly a immature T cell decide to be a B cell? Any idea for that? Why they do not do that? So these T cells, we have like shortly looked at their journey, but these T cells move to thymus, right? So in the thymus, they are still immature. T cells, immature, but they become lineage committed, that they would either die or become a T cell. So they have to work die hard for becoming a T cell, and this process is not easy. This is very difficult, like IIT JAM as well. So let's talk about that later on. But these T cells, ultimately we can see that these T cells would be bifurcated into T helper subtypes or CD8 positive T cytotoxic, right? Right? Now eventually in this course we would learn that this is not the degree of complexity we are talking about. So the complexity is way more than this. So these T helper cells, they can eventually get differentiated into many subtypes, such as Th1, Th2, Th17, T follicular. So all of these cell types have their own functions, own structures, own unique signatures. Okay, both of them, actually all of them are determined from the CD4 positive helper T cells. Okay, so the question is, how do they become all of these different cell types? Maybe some master regulator program is regulating them, right? And each of them have different type of function that we would be talking about in this course. So let me answer your question. Somehow they say are tertiary organs, right? Yes, they are actually tertiary organs. So they are tertiary, they are not primary. Okay, so that's pretty much it. That pretty much covers our understanding today on cells of the immune system. So I'll share this board with you guys. That's quite a lot of thing written in this board, right? Quite diversity is there in the board, or maybe a version of this board. So guys, do let me know whether you like this video or not. Please comment in this comment box whether it was useful, this style of teaching was useful. I generally used to teach in a different way. Then I thought, okay, why don't we do it in a old school style, in a blackboard manner, in a whiteboard manner. We can also do a chalkboard style. We have a style here called chalkboard. So do you guys like that? Or can you guys give me a vote or something, some feedback that whether you like this style or not? It's more annotated that you guys already wished in other platform. Okay, so eventually tomorrow we'll talk more about the innate immune system, and we'll talk about and practice some question based on today. Okay, so tomorrow at the same time, same day, same place, we'll be here. Okay, so stay tuned and don't forget to set your reminder. Okay guys, and you can also email me at my email address. So I'm giving you my email address. You can email me, and what we can do is form a big group such that we can sort of share all these contents, all of these boards if you need and all. So these boards might be useful, but I don't know whether whether you guys use a Microsoft whiteboard or not. Then what we can do is, like, we can share it as if a board. Otherwise I can share it with a